It's my great pleasure to introduce our third keynote speaker and the last talk of these 2024 conference. Dr. Hurley Martens trained at the Catholic University in Leuven and then had postdoctoral training in Boston at the Dana-Farber Cancer Institute before coming to Cancer Research UK in London and finally joining Imperial College about 15 years ago. 14? 15 years, 15 already, okay. Woo. And I'm absolutely delighted and very privileged that Hurdle is in the section of virology, and I'm the head of that section, but for the last 5 years, whilst I've been doing that, you've been the deputy, and it's been great to have that support. But more importantly, you've been doing some amazing work on HTLV integration and now inhibition of integration and where that's going to hopefully take us. So Hurdler, please come and take the stage. The floor is yours.
Thank you. Thank you very much, Graham, for a very kind introduction. I remember when I was a postdoc in Peter's lab, you came in one day and said, you should work on HTLV. And I was like, yeah, maybe. 2 years later, that happened. So thank you very much to the scientific committee for selecting our abstract for a keynote. I declare no conflicts of interests. And I want to start with a lay summary. So people living with HIV-1 have access to antiretroviral therapy. And they can have this to prevent disease, and we refer to this as post-exposure prophylaxis, to prevent transmission, and that can be pre or post-exposure, or to prevent acquiring HIV, again, by using these drugs before they have a risk encounter. We and others have shown that for HTLV-1, the same drugs can prevent transmission between cells in a laboratory. Yesterday we heard from Damien Purcell that it also reduces transmission in mice and reduces viral expansion in mice. We know that these drugs are safe in children and adults, so there is no reason to think why they wouldn't work as PrEP for HTLV-1 in humans. So we urgently need clinical trials. Before I start, really, I want to use this famous quotation from Isaac Newton. If I have seen further, it is by standing on the shoulders of giants. I only joined this field about 9 years ago, and I'm very grateful to all of you here and those who are not here for all the hard work that you've done on setting up all this work with HTLV-1. It's a pretty difficult virus if you come from HIV. And also very early on, there was work done to look at the early RT inhibitors, whether they block HTLV-1 transmission as well. So we've taken on the basis of the results from the giants in the HTLV field. So antiretrovirals, just to give you a short introduction to the replication cycle. So we have the viral particle. The first step in the replication cycle is the attachment to the plasma membrane through recognition of specific receptors. Following entry, so then we have fusion of the plasma membrane with the viral membrane, entry of the capsid core. And we heard yesterday also from David Jack that for HIV-1, this capsid core has also a function to prevent recognition of the RNA and then later on the DNA by the innate immune system. So reverse transcription takes place, changing the RNA into double-stranded DNA, and this is then integrated into the host genome. So this is what we refer to as the early stages of the replication. The late stages, we have progeny RNA production, RNA transcripts that give rise to viral proteins, assembly, viral budding, and then also a protease-dependent maturation. Now, if you look at these antiretrovirals that are used in the clinic for HIV patients, so we have attachment and fusion inhibitors. We have two classes of RT inhibitors, so the nucleoside reverse transcriptase inhibitors that bind to the active site of RT, and the non-nucleoside reverse transcriptase inhibitors that bind to an allosteric site. There are integrase transfer inhibitors, there are protease inhibitors, and then most recently, there are also these capsid inhibitors that block both the early stages, the binding of the capsid to host factors, but also the assembly and the maturation of the viral capsid. So these are the ones that are in the clinic and that have been approved by the FDA and also in Europe. can we use these ARVs for HTLV? In contrast with HIV, HTLV1 has a different dynamic of viral replication and viral spread. So early on, we have a lot of infectious spread. So there's this transmission of a viral particle from one infected cell to an uninfected cell. And then after a while, we mostly have mitotic spread. I say mostly because it's clear that there is still infectious spread going on. And with this mitotic spread, we have basically the division of a cell that has acquired the genomic information of the virus and splits it over the cells. Still even more. Okay, I'm sorry. So it's important to remember that these antiretrovirals, they can block infectious spread, but they cannot block mitotic spread. These are just cellular processes. So my favorite enzyme is integrase. And just to remind you of what integrase actually does, so it's a viral enzyme. Here represented schematically, and it binds in a sequence-dependent manner to the viral LTR ends. And we refer to this complex as an intosome or a pre-integration complex. The first step that integrase catalyzes is a 3' processing reaction, where it removes a dinucleotide from the LTR ends, producing nucleophilic hydroxyl groups. Following engagement with the host DNA, these hydroxyl groups attack the phosphodiester backbones, linking the viral DNA with the host DNA. And because this integration happens across the major groove, we have these gaps that happen upstream and downstream from the integration site. These are filled in by host cellular enzymes producing a stable provirus. The integrase strand transfer inhibitors, or INSTIs, that are used in the clinic block at this step, so following 3' processing and before engagement with the host DNA. Now, when we study integration in the lab, we use recombinantly purified integrase. We mix this together with double-stranded oligonucleotides that mimic this 3' processed ends of the viral DNA, and we mix this together with circular plasmid DNA. If we have concerted integration, which means we have 2 ends of DNA that it can insert it into the plasmid, we have a linearization of this circular product. If we have defective or half-site integration, so only 1 end gets inserted, we have a circular half-site product. And we can separate these on gel. So on the bottom here, it's been cut off. You would have the viral DNA. You will see it in the next gel. And here migrates a supercoiled plasmid DNA. This is a control reaction where we don't add viral DNA. Here we don't have the supercoiled plasmid DNA. And in this reaction, we have everything but the viral integrase. And here we have everything. So you can see when integrase is added, this linear product migrates differently on a gel. And we can easily measure this and quantify this. And this is where the half-site integration migrates, which is the same position of nicked DNA. Now we can do this in the presence of increasing concentration of inhibitor. We can quantify the density of these bands and then make dose response curves. And in first instance, we did this for HTLV1. We wanted to make sure it's working in our hands. And this is a dose response curve for raltegravir. So you see here percentage integration activity. And when we increase the concentration of raltegravir, this drops down. And we do this for HTLV1 integrase. This overlaps. So we went ahead and looked at other integrase inhibitors. So this is now plotted as the percentage of inhibition of integration. So the higher the concentration of drug, the higher the percentage of inhibition. So we have raltegravir, first generation compound, then elvitegravir, bictegravir, dolutegravir, and 2 other compounds. And you can see that all these integrase strand transfer inhibitors efficiently block HTLV1 integration in vitro. Now, does it block transmission in cells? And for this, we use a co-culture system where we co-culture irradiated MT2 cells with Jurkat cells that have been pre-treated with different concentrations of the drugs. Following overnight incubation, we remove the MT2 cells using magnetic beads that recognize a CD25 receptor on the MT2 cells, and then we allow the cells to grow out for 12 days, and then measure infection by measuring the proviral load. And you can see here we did this for raltegravir and elvitegravir and bictegravir. We have a beautiful inhibition of infection, and bictegravir is the most potent one. To really confirm that there was a block on integration, we also did ALU-PCR. And ALU-PCR measures the junctions between the integrated provirus with the host DNA. And this is the result. So this is the DMSO control. In the presence of ral, elvitegravir, or bictegravir, we have about 10 10-fold reduction, sorry, 1,000-fold reduction in integrated product, and on a gel, this becomes undetectable. So we did this, and we published this already in 2019. So for raltegravir and elvitegravir, we have EC50s of 6 to 10 nanomolars, which is absolutely fantastic. And bictegravir is the most potent one, which is in the picomolar range. Now, to truly understand how exactly these drugs are working, we had to solve the structure of the intosome, so the complex following 3' processing, but before engagement with target DNA. Integrase has 3 structural domains, the N-terminal, catalytic, and C-terminal domain, and all 3 of them are essential for the catalytic activity. And these structurally folded domains are linked by flexible loops, making it really an absolute pain to do crystallography with. So we first started with solving the structures of the individual domains, and we did this for the catalytic core domain. Let me go back. And the C-terminal domain. And for the N-terminal domain, we had to build a homology model because it never crystallized. And these structures are just canonical to what you observe for other retroviral integrases. Now, to form a stable complex of integrase with DNA, we spend a lot of time trying to optimize. We had a couple of things that we had to do to make this breakthrough, and one thing was by using HTLV1 integrase. So this is integrase isolated from the stump-tailed macaque, the MARB43 isolate, which is 83% identical with HTLV1 integrase. And the other thing we noticed, and this is an EMSA, electrophoretic mobility shift assay, and here we look at fluorescently labeled viral DNA, and we basically want to look for a shift. So if you have an integrase DNA complex, we would see a shift in the gel. So this is our control, donor only. Here, this is just integrase, no donor DNA. This is donor with integrase, nothing happens. We only were able to form a stable complex if we added a host factor in it, protein PP2A B56 subunit. I don't have time to go into what P56 does, but we really needed this host factor to be able to make stable complexes. We scaled up the production of the intosomes and were able to purify this by size exclusion chromatography. So you have 2 peaks here. The first peak is the integrase DNA complex, so we can measure this by looking at the protein and the DNA signal. And this is the free integrase B56 complex. Now, to make sure that we're working with a functional complex and not just the protein DNA aggregate, we could test the activity of the complex that we isolated. So we used the exactly same assay that I talked to you about before. We took these peak fractions, mixed it with the supercoiled DNA, and then checked whether integration happened. So if you do this before purification of the complex, so before injected into the column, you have a smear of integration products. If you do this in the presence of raltegravir, it's blocked. And this is the peak fraction. We again see beautiful activity, which is blocked by raltegravir. So we're looking really at a functional intosome complex. This is our negative stain EM data, 2D classes, and the envelope model that we were able to make on the basis of this negative stain. And we were really excited at this point because we saw this 2-fold symmetry, which is what you would expect if you have 2 DNA molecules that need to be integrated into target DNA. This is an overview of 2D classes. There's a lot of work that went into it, and I don't have the time to talk about it all, but we basically had to collect data on open hole grids and also on graphene oxide grids, because as you can see here, we have this preferred orientation of the complexes on the grids. To cut a long story short, we solved the structure using a combination of cryo-EM and X-ray crystallography, and found that the intosome of HTLV1 integrase is a tetramer of integrase. We have 4 molecules of integrase, 2 molecules of viral DNA, and 2 molecules of B56. And I'm not sure yet it's going to work, so this is just a video made by Michael, the postdoc, who did the work. So we have 2 molecules of viral DNA, 2 molecules of B56, and 4 molecules of integrase. Now, at the same time that we were doing this for HTLV1 integrase, there was a group who did the same thing for HTLV1. And they used a completely different method. They also needed B56. They assembled it completely differently, but we come to the same conclusions. Our structures overlap fantastically well. So we're really pleased to see that. We went ahead to study how some of these compounds bind to the intosome. So we optimized our endosome assembly for cryo-EM studies. And we did this now in the presence of either raltegravir or bictegravir. And what you can see is here we have superimposed the active site of our structures of HTLV with raltegravir with the structure of previously solved foamy virus integrase that binds to raltegravir. And here, this is superimposed on HIV structure bound to bictegravir. And you can see that the binding of this integrase transfer inhibitors is very similar. And this also explains why these compounds are so potent in blocking HTLV1 integration. So when we just finished that work, there was a long-acting integrase inhibitor that was approved by the FDA. That's cabotegravir. And we also checked whether this is able to block integration and transmission in vitro. And this is the work done by Bethany Schneiderman. And we can see that in the presence of cabotegravir, we have a complete block of integration measured by ALO-PCR. So when I presented some of this work in Brazil on the HTLV symposium, I believe it was Carlos Brites who asked me, so what about dolutegravir? Have you tested dolutegravir? And I said, well, at the time, we didn't check it in our in vitro, in our co-culture system, because there was a scare that babies born from pregnant women who live with HIV, that they were to have neuronal tube defects. Fortunately, this was a scare, and it's been shown that dolutegravir is safe to be used in pregnant women. So we went back to check in the lab whether it's able to block infection. And this is a work that Mphatso also presented on Monday. So here we see in the presence of increasing concentrations of dolutegravir, we efficiently block HTLV1 transmission. This is measured by proviral load with an EC50 of 0.25 nanomolar. And as you can see here, the cells are very happy at these concentrations of drugs. And this is measuring ALO-PCR. So this is 80 picomolar concentration. We have about 60% reduction of integration. And at the higher concentration, 800 nanomolar, we have a complete block measured by ALO. So this low EC50 was not that surprising at the end, because the structure of dolutegravir and bictegravir, which was the most potent one so far, is very similar. We had previously tested whether tenofovir desproxyl fumarate blocks HTLV1 in our tissue culture system, and it does work. It's not as potent as integrase transfer inhibitors. So we also checked the first-in-line drug TAF. And as you can see, we have a beautiful dose response curve, EC50 of about 12 nanomolar. And we haven't solved the structure of HTLV1-RT, but Mphatso did make used AlphaFold to make a model of HTLV1-RT. And as you can see, the active sites look very similar, but most importantly, the residues that are involved in binding to TAF are 100% conserved, again explaining why this drug can block HTLV1-RT. So far we had worked with Jurkat cells. The question was, can we reproduce this in primary cells? And this is work done by Alison Maher, a PhD student, a former PhD student in the lab from Margarita Dominguez-Vilar. And here, so we used the PBMCs isolated from blood donors and checked whether in the presence of tenofovir, raltegravir, or dolutegravir, we can see a block of infection. And this was measured by TAX immunostaining. And FACS, sorry. And you can see that in the presence of the higher concentration, we have a complete block. And if we use the drugs at the concentration of the inflection point of the dose response curves, we still see about 80 to 90% reduction in infectivity. So let's have a look at all the drugs that we tested. So we have the IC50s, which is the inhibitory concentration to block the in vitro recombinant protein in the enzymatic assay, and EC50s is for our cell-to-cell infection system. Now, if we compare our EC50s with the EC50s that have previously been published for HIV-1, you can see that they're almost identical. And the same is true for the NRTI inhibitors. They're actually even, TDF is slightly more potent in blocking HTLV1 infection in vitro than it is in HIV. So all our tests at INSTIs and TAF-DDF block HTLV1 transmission in vitro as effectively as HIV-1. We show that RAL, dolutegravir, and TAF also efficiently block HTLV1 infection in primary cells. So there's no reason to think why they wouldn't work in PrEP. And yesterday, we saw this really interesting and promising data, and this is not by me, this was shared, graciously shared by Damien Purcell, and it's the work done in collaboration with Marcel Derflinger and Mark Pellegrini, where they used humanized mice. And they showed that in the presence of tenofovir, either prodrug, but not 3TC, which is HTLV1, has a natural resistance against this NRTI. You can see that there is a reduction in transmission of HTLV1C. And when you combine dolutegravir with tenofovir, there is a reduction of the viral spread. So this is really promising. This is the first animal model data where we can see that these antiretrovirals can block HTLV1 infection and spread. I was really excited to see the work from Carlos earlier on in the conference and this morning as well, where they looked at people who have been on ART and specifically also integrase inhibitors, that they could show that over time there is a reduction in the proviral load of these patients, and that is correlated with the length of time of therapy. So again, really exciting for the future of the antiretrovirals with HTLV. So if you look at all the different classes of antiretrovirals that we have, so the entry and fusion inhibitors that target human CD4 and CCR5 are unlikely to work for HTLV1 because it binds a different receptor. For the NRTIs, so some of them work, not all of them. The allosteric inhibitors and non-nucleoside reverse transcriptase inhibitors, they are not going to work. There's too low sequence conservation between HTLV1 and HIV1-RT, so we need specific compounds. All of the tested integrase inhibitors work. For the protease inhibitor that's in the clinic, there's data showing that there is no or weak inhibition and that we need HTLV1 specific compounds. And it looks like that both Celia Schiffer, Louis Mansky, and David Jack are going to hopefully come up with some exciting new drugs in the near future. And then the lenacapavir, which is a long-acting HIV1 capsid inhibitor, doesn't work. We've shown this, and also David Jack has shown this. It doesn't block HTLV1 infection, so we need specific compounds. But there's still a lot of open standing questions. We don't know whether HTLV1 will develop resistance when there is pressure of ARVs, prolonged pressure of ARVs. And this is something we have to be careful with, and we have to think about this. Can we also develop these long-acting capsid inhibitors? Can we optimize the protease inhibitors? We should also look at novel targets of antiretrovirals. One class of inhibitors that is in clinical trials for HIV1 is an inhibitor that blocks the interaction between HIV1 integrase and a host factor LGF, so really promising as well. We haven't tested whether these inhibitors are effective against HTLV2, but it would be very unlikely that they wouldn't be because they are so highly conserved. So the future direction, we need clinical trials. We need both retrospective and prospective studies to answer the question whether we can block sexual transmission of HTLV1, whether we can prevent mother-to-child transmission, and can we block infection from organ donations, blood transfusions, and non-medical exchanges of contaminated blood. So we started this conference with, and I think we all felt indeed what the patients were saying. And I agree with Anne van den Broek who said, I'm scared. But I think at the end of this conference, we can also be hopeful. And I was really excited that we had so much support for the petition that Fabiola and I made to draw attention to and put pressure on the pharmaceutical companies. Why do we not eliminate HTLV1 whilst we are eliminating HIV1? So we had 937 signatories from all over the world. We had signatures from clinicians, academics, patients, and the community. And I think it's amazing that we all come together as a community with all our different backgrounds to put pressure and to start thinking about also ourselves setting up clinical trials. So we talked a lot about collaboration during this conference. So I think it's time we set up an HTLV PrEP clinical trial working group. We've got the stamp of approval from Fabiola as the president of the IRVA. Graham Taylor is also on board, I'm really excited to say. So the aim is to eventually set up clinical trials to see whether we can prevent HTLV1 transmission using these ARVs. The terms of reference still need to be determined. I'm asking you, are you interested are you working with in an antenatal clinic? Do you have access to patients? And really important in this study will be also to look at the follow-up, so to follow the neonates that have been born. We also would like a patient representative to be present in this working group so that we can come together and decide and make a work of developing clinical trials for this. So if you're interested, this is my email address, and do get in touch. And then at the end, I just want to thank all the people that have been involved. Patso, Bethany, Michal, Jordan, Teresa did the work that I presented to you today. I'm grateful to the rest of the lab. It's always very interesting to discuss and get feedback from everybody. The CRIEM was on a collaboration with the Cherepanov Group and Nora Cronin from the Lonsem facility. The open letter, Fabiola, our working in different time zones, is at the same time very intense and very exciting. It's been fantastic working with you. And the other members of the team on the open letter, and of course also Margarita and Alison, and the people who donated the blood for us to do the studies in primary PBMCs, and the funders. Really quickly, you've probably noticed Reciprocal Space. This was a company set up by Michal Barsky, so the guy who solved the structure of the HTLV1 intosome. He was a postdoc in my lab. So he set up this company together with Yatsil, you have seen here over the past few days. They're doing web design. They did it for the conference. They also did my personal lab web design. They're fantastic. So if you need something, go to them. And then finally, there is a special issue in pharmaceuticals from MDPI, and the submission deadline is the 30th of November. So if you have presented any data that you're still wrapping up and you want to submit a paper, please do so. Thank you.
We've got time for a couple of questions if anybody has a burning question.
Yeah, beautiful talk. And taking it to public health is really interesting and important. So my question is related to protein phosphatase 2A. Do you think that is somehow altering integrase activity, or vice versa? Is the integrase somehow altering PP2A activity in the cell? Okay.
So we know that for integrase to bind to PP2AB56, it doesn't have to be modified, because this is recombinantly purified protein coming from E. coli, so there is no phosphorylation necessary. Whether it dephosphorylates at this stage, waiting for mass spec results. Whether integrase is interfering with PP2AB56 function, I would say it's very, let's just look at it from the number of integrase molecules that we have in a virus that comes into the cell and the amount of PP2AB56 there is in the cell. So we have about 100 integrase molecules, and we have thousands of PP2AB56 molecules. So if the question is whether integrase can block activity, for sure. Is it functionally relevant, biologically relevant? I don't think so.
Okay, thank you very much. We're right on time. Thank you, Herder. That was a great talk. Thank you. So I'm probably going to confuse things now by holding the mic. So we're now going to do a feedback from the workshops. I hope you all enjoyed the workshops. I was probably the only person who went to all 6, but that did mean I was dipping in and out of them. I thought it'd be really important to get some feedback, so there's cross-fertilization from the different workshops. And I've asked one of each of the workshop chairs to come up now and very briefly to talk about the sort of main points that came out of the workshops. And we're going to do them kind of in the order in which they appeared on the program. So we'll do the left-hand side of the program, if you like, first. So we're going to do 1, 4, 2, 3, 3, 6, 2, 2, 5, 3, 6. Yeah, something like that. It's the end of the conference, in case you hadn't noticed. So we'll start with workshop 1, please. And that was Amanda and Pat. Great.
I'll just say a couple of words. So Graham got in touch with Luke and I about a week before the conference and asked, the way these workshops worked is, asked us to put together a program we got together on a Zoom call with some of our colleagues of other workshops. Ann Vandenbroek and Damien also participated, thought about what the approach might be, and we talked about it a lot and figured out how we were going to approach it. We decided that we would ask Amanda to sort of lead off, Dr. Amanda Panfield to lead off and give a bit of a summary, but our goal was then to look at other abstracts that were submitted to the meeting that were interested in doing orals, but because of time we couldn't, and so we sort of picked abstracts from that, and those were the ones that gave talks at least our workshop. So I'm going to now turn it over to Amanda.
Yeah, so as Pat mentioned, really in the introduction, we just kind of gave a broad overview of the available animal models in the field. So, you know, we have BLV as another Delta retrovirus, and we can learn a lot with comparative virology studying BLV. And then we have a sheep model to mimic, you know, BLV-induced disease. It's accelerated. We have various different non-human primate models. And then we talked a lot about different mouse models, right? We have transgenic, immunodeficient, depending on the way you inoculate the immunodeficient mice. Where do the tumors form? Is it static? Is it metastatic? And then humanized mice and really the advantages and how that's propelled the field forward. We discussed a rabbit model of persistence and infection, and importantly, I think we really discussed that we urgently need a model for ham-TSP. We do not have a model for that, and I think that is one glaring hole in the field if we want to keep studying HTLV diseases. And as Pat mentioned, we heard a lot of very excellent talks, which I won't have time to highlight here, but we also talked about the development of an HTLV-1 vaccine. So I presented some of our work looking at how we developed an envelope mRNA lipid nanoparticle vaccine and tested it in rabbits. And that brought up a lot of interesting discussion. And so some of the highlights that Damien very nicely summarized and shared with me, his scribbles from the talk. So hopefully I've captured it all here. But really, who are going to be the users for this vaccine? Is it going to be everyone? Are we going to focus on endemic areas first? These are just all things we need to consider as a field. Communicating with those users. A lot of these people are from indigenous communities, and we need to talk about the acceptance of those vaccines. Are we going to look at neonatals and try to block mother-to-child transmission? Can we even piggyback off the acceptance of other vaccines in the region, combining different vaccines together to get public acceptance? I think the big thing is who's going to pay for this? Who's going to be the customers? Is it going to be individuals? Is it going to be a government that's going to fund this? What are going to be the benefits? That's how we're going to have to sell this. Is it going to be an economic benefit to the companies? Is it going to be a social benefit? We need to sell. It's going to be a profit for the developer, so we really have to sell that. What's the market size? How many people are going to require this vaccine? What are the clinical trials going to look like? How many people will we need to show efficacy? I think Luke touched on this when he was describing the BLV vaccine during the talk, but manufacturing feasibility, what is that going to look like? And I think that's going to be what type of vaccine is going to be the first one that we can bring to market. You know, obviously mRNA vaccines, you have that cold transport. That's going to be a problem in resource-poor countries. But if you look at other types of vaccines, maybe those are better for those regions. And then I think this one's really important too, but what are the measurements of success going to look like for an HTLV-1 vaccine? So are we going to decrease severity of disease? Are we looking for the unattainable sterilizing immunity? See if he looks up. No? Okay. Are we going to decrease proviral load? What is going to be the measurement of success? We need to really come together as a field and decide that so we can set what our goal is. And I think the other thing that I took away from this, at least me personally, is we need to unify as a community to accomplish this. It's not going to be one vaccine and one group that accomplishes this. It's going to be sharing the knowledge, sharing the resources that really make this a doable concept. Yeah, thank you.
Okay, so for the workshop 4, we were over 100 people. So, well, despite the large number, I think there were really good discussions. I think people asked a lot of questions. We had good discussions. So I will present a part, and then Damien will follow up. So first, we had some workshop themes, and I will talk about 2 highlights that I think in the genomic technology session. I think that despite this very small proviral genome, I'm amazed, like every time we meet, there's new things that are discovered in these small 10 kb things, like these novel regulatory regions. We know more about CTCF now, the OCR silencer region, but also in HTLV1C. So a lot of defective provirals, what does that mean? It's important. And then VEFA's group with the P16, which is acting very differently compared to what we see in A. So I think that, again, this proviral genome is still surprising. And this was one of the points. The other one is the extracellular vesicle session, where actually we learned that EVs are involved not only in cell-to-cell transmission, but only look like drive immune modulation. So at the end of the session, we asked 2 questions to the people and asked a feedback to us for these 2 questions. And so there was a kind of good feedback. And the first question that we asked was, what are the greatest scientific barriers to understanding HTLV1 infection and disease? And so the feedback we had from many people is lack of funding, lack of funding, lack of funding. Private, public, commercial, for basic research, but also for translational. Once you start to go to translational, we have a rare disease, so it's really hard to get money for that. It's expensive. I think we need international consortiums. That was also feedback that we got. Also, what we hear is difficulty of publishing, asking for kind of reviewing to facilitate further applications, but also further grant submissions. People still think the field is not prioritized, and there's still a lack of awareness, even if we really push for it. And then someone asked about, really the lack of testing and lack of access to samples for basic scientists. So we need to increase the interactions with clinicians. So the second question is for Damien.
Thank you. Yeah, so the second question, we're asking about what scientific development would deliver the best outcome for people living with HTLV? And very clearly, treatment for HAM-TSP and treatment for ATL. So neither is effectively treated now. So further understanding the targets and mechanisms to achieve that, be they immune mechanisms, we've seen drug targets, new concepts coming through. We had some in the sessions in the workshop, and particularly during the meeting here, we've seen lots of interesting things. So questions coming forward also were, what are the causes of spontaneous remission in ATL, and how frequently does that occur? I think it's pretty important to look at these offbeat cases. We've seen some of those things coming through, and when we find those things, they can really give us great insights. And these can answer questions suggesting new therapies, particularly for ATL. And then importantly, looking at other mechanisms for immune function. So we heard Vefa talking about the importance of monocytes and NK cells and CD8 T cells, depleting those and seeing that that then permits very rapid infection. But mechanisms underlying this, like Vefa was mentioning, efferocytosis, we saw a nice talk on that, and the role for this new P16 protein that she's found in the subtype C, indicated from earlier work, but here it is something that's potentially impacting on this mechanism that we don't really think about that much. Similarly with NK cells. NK cells have got a lot of potential capability for immune activity against HTLV. And then also other mechanisms that we are getting a glimpse of, for example, seeing these very high rates of provirus deletion, even in animal models like the human immune system mice, where there's no immune, T-cell immunity, yet very rapid acquisition of deletions. We saw a nice presentation in the workshop from Natasha Jantz about this. Mechanisms maybe involving things that we'd mentioned briefly, like the Hush complex, things that involve cellular proliferation, DNA damage. Can we actually drive that process so that we drive the virus into non-transmissible state by accumulating deletions and rendering it non-transmissible, and maybe even driving it further to delete HBZ, TAX, and eliminate pathogenesis, so just leaving a scar that is not capable of sustaining pathogenesis. So there are many interesting mechanisms, things in immune function that we haven't yet accomplished, like making HBZ, a potent T-cell antigen, something that Charles is suggesting. I think that's a great concept. Can we drive immunity harder towards achieving outcomes in terms of eliminating pathogenesis by immunity? So that was our workshop, and thanks to everyone who attended.
So myself and Georgie and Karu,
international guys,
hosted a workshop concerned with the prognostic tests for asymptomatic carriers. So shall I? Yeah. So first of all, we reviewed the state of research. What's our understanding on the possibilities of predicting the outcome of HTLV1 infection? And it's really great that we've got clinical and laboratory evidence that can predict the development of ATL and also HAM. But this is critically by detecting early signs of disease activity. Something to think about is that in order to detect the onset of early onset of disease, is that we need continued surveillance to catch these early signs. And our consensus together and the group as a whole was that proviral load measurements are very, very useful in determining who is at risk. However, this assay lacks specificity, and asymptomatic carriers may have results that overlap with people with disease, so it can be very difficult to predict just on the basis of proviral load assays. And there are some massive gaps in our understanding, like if we see these early signs of disease, when will disease symptoms onset, and what fraction of people in whom we see these early signs of disease will remain healthy? And we've known nothing about predicting the risk of developing other rare HTLV-related diseases, and very little understanding of this all-cause mortality that's associated with HTLV1 infection. So huge basic research questions to answer there. Reviewing, globally, or well, at least whoever was in the room from around the corners of the globe, actually, though we know that this proviral load assay provides useful clinically useful information, there are very few centres around the world that are able to provide proviral load testing, and there are lots of reasons for this. One resounding thing that everybody agreed on was that it's too expensive, and often insurance or national insurance doesn't cover this test. Other prognostic tests that we discussed that detect evolution of malignant clones or signs of inflammatory diseases. These are only available really in research settings now, or a very small number of centres. So we've really got to think ahead as to how we can make these available more broadly. So, looking forward. So we really need to push for a commercial proviral load test, for example, on some of the platforms that are used in diagnostic labs for high throughput testing across the globe. And we absolutely have to find ways to reduce costs. So we need to think, I think it'll be up to us to think of ways for perhaps maybe considering different sample types that require less hands-on time in order just to get that cost down so these tests are more accessible. We absolutely need to raise awareness within government bodies, clinicians, and the wider community to push for better provision for carriers. And I'm so happy that this testing working party has been established. And during this meeting, we decided to set up a subgroup within this testing working party that's going to be focused on prognostic testing and thinking ahead to how we can provide these tests that we're seeing are quite useful in research setting and making them available to the broader community as well. So knowing the outcome of disease or being able to detect disease very early has the potential to change care. So that's going to raise, going forward, the need to consider guidelines and develop guidelines for clinicians to think about how to discuss risk with patients, whether to intervene or not, and when, and what treatment. And I think there's a huge research gap there as well in developing new treatments that are acceptable to give to somebody who's currently healthy, that we can see these early signs of disease. And I don't think we have a perfect drug, at the moment, but we should test what we've got. But fundamentally, this should be guided by the people who are living with HTLV. How asking them, how do you feel about taking a test that might show you have early signs of probably fatal or potentially severely disabling disease? And if you were healthy and you found out you had early signs of disease, would you actually want to participate in a clinical trial? So taking their guidance really on how to proceed here. So I think that's all from us. If you have anything to add. Yes.
Great. So Raymond and I chaired the session on international collaboration, which was a great session. So we designed the session by considering some of the topics that have been listed in the WHO technical report and that highlighted gaps, because we thought that perhaps that might be a good starting point for gathering data to help kind of inform public policy. And we did this by having a couple of talks. We reviewed some abstracts which were presented. We had some discussions, and we did a little bit of voting too. So we started the session with an overview by Cédric Contaré, who is a public health doctor who works in the field of international collaboration. And he has produced these kind of fantastic maps, like at the bottom here, where he could look in sort of previous decades as to who was working with who globally in HTLV, and you could kind of refine it by topic, by subject. And you could see how it's evolved and how, in particular, Brazil has boomed in the last kind of 10 years in terms of what they've been publishing and doing. And but then he introduced us to something called DOSCAR. And I would really urge you to have a look at this website. The website is doscar.com. And he has established, this is the first website dedicated to clinical and scientific research and collaboration in the Caribbean. And you can have a look at the website, but you can look island by island. You can see who is publishing on what in whichever field. You can see, it's a fantastic tool for bringing together people who want to work in healthcare or collaborate in healthcare in the Caribbean. And it's all the islands, not just the French Caribbean. And then, there we go. And then Antoine spoke to us about ANRS, which sounds like a fantastic French, I don't know what you would call it, like a public health body, but it is focused on the facilitation, coordination, evaluation, and funding of research. And it has patients at the center of all of its missions, which was just great. And Antoine is really focused on setting up inter-regional collaboration and including all of the Caribbean and Latin America. And it would work very neatly with the DOSCAR proposal. And then we had a whole range of talks that covered some of these areas. So as per the other workshops, the need for novel diagnostics, and Harris, who I'm sure some of you saw his poster on commercial diagnostics. We talked about the blood service and what we were doing well and how to look after blood donors and how we could share experiences. We also talked about the vaccine cohorts in Brazil. I talked a little bit about some of the challenges in ATL and where we have gaps. And then Adine spoke about the UK Patient Forum, which is quite well established. And she gave some perspectives from what patients want from research in the UK. And then we asked people before they left to write down what they thought that their kind of top priorities were, having heard this kind of mismatch of different subjects. And this is roughly what came back. So there were more than 60 comments. Some of the comments were kind of what I would say was cross-theme. So, the need for different, endemic versus non-endemic countries to work together, raising awareness. Can we piggyback onto the back of HIV and hepatitis trials? The need for large cohorts, but also control populations for cohorts. Regulatory issues and patient advocacy being weaved through everything. This, obviously, we've had time and again, and it's clearly the big thing for this meeting is the lack of availability of diagnosis, really basic need for serological assays in many parts of the world, assays for ATL, pre-ATL, and pre-HAM. And really that there's a lack of guidelines, and that's guidelines for diagnostics. Asymptomatic carriers, there is not an international guideline how to manage an asymptomatic carrier with HTLV1 infection. Pregnant women, pediatrics, and of course, and how to manage a blood donor. Big need for prevention, we've just been discussing, intervention studies, and therapy and management. And actually, we talked a little bit more about patients and how do we give them information, what's the psychological support around this, and there's probably really not enough understood about the best way. Anything else? No? Okay. Great.
I think, Lucy, you gave us a very good introduction because a lot of what you talked about was also in our workshop on elimination of vertical transmission. And, we basically know that there are a lot of really unanswered questions, and also the previous group who talked about testing, getting the right tests, simple, straightforward diagnostic pathways. So much we don't really understand about the pathophysiology of transmission and how does that really happen, and what makes you high risk and low risk. And is that the virus, is that the host, or the combination of both? And then a really big issue, I think, is level of knowledge, which includes healthcare workers and the general population. And we need to get out there and educate our colleagues in particular about this virus. And then thinking about interventions and how do we risk assess our pregnant women. What does it mean to be avoiding breastfeeding, the dangers of that in certain situations, whether or not cesarean section has an addition, and of course the role of antiretrovirals. And it's very interesting because in the field of HIV, where I've spent most of my working life, pregnant women and babies were always at the back of the queue. It takes almost 10 years to get the dosing for neonates for an antiretroviral, and adults will have had it already for many years. But actually, with HTLV, pregnant women and babies have the chance to be at the front of the queue because we've done all the pharmacokinetics, we know all the safety data, so that's actually quite exciting. And of course, is there going to be a role for broadly neutralizing antibodies? That's a big question. So I think even with the limited data that we have, what can we do? And lots of lessons to be from HIV. And we talked about the fact that we don't have guidelines, but in the UK, we are actually trying to make some kind of a risk assessment of who's low risk, who's intermediate risk, who's high risk. We have actually made a kind of algorithm that we think is reasonable to consider that takes into account viral load and also symptomatology and what we would want to do in terms of antenatal, perinatal, and postnatal care. But of course, that is based on no randomized control trials. So what do we need to do now? We know what we can do to prevent transmission. We know what makes a difference in certain situations, but we definitely need more evidence. We need the simplified diagnostic pathways. We actually need international surveillance. We need to think about the randomized controlled trials. We need guidelines and good practice, and we need access to formula and clean water and antiretrovirals and education. We don't have international prospective cohorts of infants born to women being followed up, whether they are HTLV positive or negative. And these are all things that would really be helpful. What was fantastic was to hear from our Brazilian colleagues about what they are doing and what they are taking forward. In 2024, they will start to test pregnant women for HTLV and will start to implement interventions. So we will have a huge amount to learn. Very quickly, we will have hundreds of babies born to women with HTLV. So that will be really important. And we also were very lucky to have Meg Doherty in the workshop, also Leandro from PAHO. And the WHO, as you know, has put together its plan for triple elimination of HIV, syphilis, and hepatitis B in terms of vertical transmission by 2030. And I think what we need to be thinking about is making that into quadruple elimination. Thank you.
So that's the last workshop that we held on Sunday. Thanks again for the organizers and for the participants. It was a workshop on how to prevent the transmission of HTLV sexually. As you know, about 70% of the transmission contribution is sexually, and about 5,000 sexual transmissions have been recorded in Japan every year. So what we looked at were the workshop style was 4 lectures followed by 2 workshop groups, Group A, Group B. The 4 lectures were delivered by Meg Doherty and Leandro Serrano from PAHO and WHO, and then Angelica Mirna from the Brazilian Ministry. Then we had Pip Hetzel, Director of National Retrovirology Lab in Australia on notification, and then a summary of data available on sexual transmission of HTLV by Daniel Bradshaw. I thank you all very much for coming and presenting to us the latest data. Then we had the 2 groups. The 2 groups focused, one group focused on HTLV testing and notification, what the challenges were, what the processes were in different regions, and then how and what should be addressed or improved. how barriers removed. And the second group focused on the sexual transmission, again, what was available in which regions and how these barriers could be removed and what should be improved. And in summary, the WHO was very open about the fact that they have promised a lot and not delivered enough. And Meg Doherty and Leandro talked to me about the 12 recommendations and that they're very keen to continue working on it, especially this year. And IRVA will support them, but each and every one of you is most welcome to contact them directly and support them. So they were very open about this, and I'm really grateful for their presence and for their honesty. Secondly, notification. We are really proud and guided by PAHO and by Brazil. Brazil has now established, formalized the fact that all women in Brazil will be offered antenatal care testing for HTLV. This is 3 million deliveries a year. That is a huge progress, and I'm really proud of Brazil for this during a very difficult economic and political situation that they have been in for years. So fantastic and well done, everyone. And we will also learn from you about how you notify and how you keep notification going. And what is really important about the notification, the workshop said that the notification and surveillance has to be established in every and each country for us to really know where HTLV transmissions occur and what the incidence and prevalence is. And as you know, and you know from the ECDC papers published by Geosciences, we just don't know what's happening. So we really need you guys to make HTLV notifiable. I'm looking to PIP for Australia. We need to do better. We have only mandatory notification in Northern Territory and not in the other 7 states. So please, if this is the case where you are, try to make a difference. HTLV testing, we have tests. I'm a clinician, but they're not good enough. We need better screening, and screening, I think we're okay, but we need better confirmation, and we all agree that we want it easy, affordable, accessible, and we like PCR more than Western blot and blah-de-blah. So this is all I have to say as a clinician. Please give me a point of care, home testing that gives me an antibody antigen test so that we can just roll it out. And Japan is a forerunner for the pocket test, so well done to Japan. Please commercialize it and make it accessible to us. Now, the sexual transmission, here we have 4 points to make. And we were very good at bringing everything together. We will not be able to make much progress, no matter what we do, if we do not destigmatize and take away the shame from having a blood-borne virus, a communicable virus. It's just a virus. It doesn't discriminate, nor should we. So we need to work really hard on destigmatization of people who carry the virus or catch the virus or transmit the virus, so that they come forward, they can seek help, and they can be tested, and their families and partners can be tested. Then we need health education and awareness globally, regionally, statewide, to everyone, so people know what this virus is. We need a very efficient, effective education of the healthcare force. Globally, this is not available. Again, WHO could lead on that, should lead on that, call on them to lead on that. And we would need to have, we want to look after the different groups of people. So testing is another thing that we discussed, meaning offer testing, make testing really easily available to at least certain groups of people. Antenatal, we discussed, roll it out. And then also in sexual health clinics, roll it out. We have discussed cases where patients show up at STI clinics in this country. We are really good at having STI clinics still. And they want an HTLV test, and they're sent away by specialists like myself. And that's absolutely unacceptable. So we need to be able to offer testing not as a general screening for everyone and everywhere, but at least in specific clinics, antenatal, sexual health, renal transplant, blood donor. We need to be able to test when people ask for it. And then finally, we need to look after those people who have HTLV, and they have to live with it, they may want to have babies, so we need to support them to plan their families. And then we need to also support those who just want to have sex, because believe it or not, it's part of being human. So need to have sex to feel free and happy and safe from not transmitting it to someone, and also for not being criminalized for having possibly transmitted it unknowingly. So then that's where Gödele and Damien's lab and other people come in. From VEFA, we need vaccines, we need PEP, we need PrEP. And in the meantime, we have PEP and PrEP available to us. Repurpose the drug, write in your notes, offered off license, and support your patients. That's it.
Thank you to all the speakers. I think that's an amazing summary in the last half an hour of a huge amount of work. And I'm going to propose that we take the transcript and edit it slightly and then produce a document from this conference for IRVA. If that's, yeah, okay. You'll all get a chance to make sure that you're quoted properly. But I think that would be an amazing output. Okay, Fabiola, don't go away, because we're now moving on to the awards. Obviously, there were awards announced at the AGM yesterday. Was it yesterday? It was yesterday, wasn't it? Yeah. Yes. But we've got other awards. So, Fabiola, if you'd like to come out and join me.
We have no awards. You will. Right. Let us start with the beautiful. Let's do this at the end. Now, Graham, I think you have certificates for travel awards. Would you like to do that? Because your team chose the travel awards.
Thank you. So, for the organizing committee, it was really important to get as many people as possible coming from the low and middle income countries, and we provided funds for those people to join and register for the conference. Viruses from MDPI also gave us 2 travel awards, and we had a look at the people coming from low and middle income countries and selected on the basis of the highest score of their abstract. So I'm really pleased to tell that Iris Montaño Castellón has awardee, and this is Renata Olivia Cadena Romero.
Okay, so we presented you the IRVA International Award recipients yesterday, but they couldn't come because they were doing exactly what they were supposed to be doing, promoting IRVA and the HTLV and everything around HTLV cause and the patient's needs at the Brazilian embassy. So HTLV channel Thank you so much for all your good work. The people have chosen you as the awardees of the 2024 IRVA International Award.
For your amazing work. Well done. Now we come to the final awards.
So now we came to the time of the poster awards, which was very difficult to make a classification because there were 120 posters. Most of them were present here. And so we have a lot of discussion and vote. And so we decided to give the poster award to Aurélie Beauvois. Okay. So, maybe she will. Maybe if you want, you can, in 10 seconds, say a few words of your poster.
Why don't you read the title first so you can catch your breath? Come here.
The title is Helicase-like transcription factor restricts the infection cycle of HTLV1 but interfere with T-cell mediated antiviral cytotoxicity. I'm sorry.
What's your work about? Just tell us. Sure. It's his idea, not mine.
So in my lab, we found a protein that restricts HTLV1 infectious replication. And so when we upregulate this, the expression of this protein in HTLV1 infected cells, infectivity of HTLV1 is reduced. And at the same time, this protein also interferes with T-cell cytotoxicity. So it's a restriction factor, but it also enables escape, immunity evasion.
Fantastic. Thank you so much. So you know what's coming, the other two, but you just don't know who you are.
So, so the second winner is Eric Liu. Is it here? What? Eric Liu. Yeah, very good. And the title of the poster is North American Adult T-cell leukemia lymphoma patients has frequent mutation in CCR4 and respond in vitro to small molecule CCR4 antagonists. So
so what am I supposed to say?
You can, you don't have to.
Okay.
You can tell us a little bit about what it is.
Hi. So my poster was way back in the corner, so I didn't expect to get called upon. I just joined this field within the last year or so, and we have a unique opportunity to study with a large Afro-Caribbean population in our surrounding hospital region. I think this is a really interesting project, a really good, conference organization, and this is, my first time presenting a poster at a conference, so I'm really excited.
Is this your first poster?
More or less, yeah.
Wow. Junior.
Now we have a very young recipient who is Damien Purcell.
Very, 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 very shy.
So maybe you will talk about the vaccine, the progress towards the vaccine in
So
HTLV1.
he scored top on relevance. Well done.
Well, thank you. Well,
Firstly, I have to thank the members of my lab who actually stood at the poster and presented it while I was marking posters. So my poster was about development of mRNA vaccines in my lab, where we're looking at mutations to stabilise the trimer and also to incorporate that onto virus-like particles, as well as expressing high levels of protein so we can determine the structure of the HTLV trimer. So thank you very much. Totally unexpected. Thank you. First poster prize I've ever won.
And then this year we have a special poster winners coming from the vote from the Patient Association, and they decided to give the best poster to Ingrid Burkhardt from London. She's here. The title of our poster is Timing of Loss of Maternal Antibodies in Infants Born to Mother Living with Human T-cell Virus. Oh, it's you.
It's also the youngest recipient of this. And the member is the youngest member, and we have you. The printer has failed, so we'll get you one. But please, if you have some words to share.
Yeah, thank you very much. 2 months old, youngest presenter. Yeah, so we have a very small cohort of infants exposed to HTLV through their mothers, and we followed up their serologies, and we sort of saw that they all got negative fairly early, about 18 months, which might sort of change practices around how we can screen them and how long we have to follow up. And the shorter we can do that, the more likely we're not losing them to follow up, and also sort of can give parents and their babies the clear and to live sort of without the burden of HTLV.
I have no idea. Let's almost.
We can do it
We'll try and do it. No, not yet. Not yet. I think we have to go back. Yeah. Okay. So here we are, the end of. It seems like months since we met on Sunday morning at the closing session. I think you've enjoyed it. So very sad to be at the end of the conference, but I have some good news coming up. I just want to highlight again the support, the organising committee and the scientific programme committee and others which I'll come to. I just want to tell you how many people have come up to me and said what a good conference it's been. And it is a team effort. So many people have been involved for so long, and I want you to give your appreciation first to the organising committee. I was because your hands are going to hurt. Scientific Program Planning Committee. And then to our patient reps and their supporters. And, I've lost my, anyway, I started with this slide about the support we had, and I mentioned many individual people. I didn't mention the HTLV channel. I thought they would be doing their own publicity. They did. You've seen them around, and they're doing great work. But I also wanted to thank the Royal College of Physicians. I think the staff here have done a great job. Thank you, tech guys. They have to do a lot louder to get through to the catering staff. And if you need them, if they're helpful at all, there are 17 hours or 17 CPD points available from the RCP, so you just put it down in your continuing professional development if that's something you need. They're all there for you. It's all registered. And then, of course, I want to thank MedEvents. The team outside, Karen, Gemma, Glenn, and Nikki, and Maggie, who was with us before, she went off to have her baby. But they've been tremendous throughout the 2 years that we've been working with them. And you haven't heard the last of them yet because there's a survey coming your way, which I really hope you'll engage with. And I know we've asked some questions, but I think actually I'm really looking for some free comments about what worked, what could have been done better, and thoughts about the next meeting and how that could be designed as well. So please look out for the survey, please respond, and give your thanks in the usual manner to MedEvents. That bit we've had. Now, I think hopefully, yeah, there's a really, well, there's a really important group of people. Next slide, I'm going to move on, yeah. I've been totally impressed. I mean, I'm still impressed. I'm looking at a room that's almost full. It's 3:25 on the fourth day, and I didn't see anybody asleep in the entire.
You were sleeping.
In the entire conference, and I've taken photos on several days. Keep thinking there'll be. I just got to catch the room whilst it's full. And then I come back the next day and I'll take another one, it's still full. So thank you so much. I'm really impressed because we mixed up everything quite a lot. We've given you everything from basic science and virology and molecular biology all the way through to policy. And I think everybody's pretty much been here for everything. And I think that's a record for any of the conferences that I've ever been at. So I want you to give yourselves...
Wait.
Oh.
I have something to say. I've got feedback from MedEvent about you guys.
Oh, go ahead.
They were very specific. They said, you're a civilized lot. I loved it. I said, compared to the surgeons? I said, yeah. And then they said, you show up, you go in when they ring the bell, you stay for the posters, and you're kind and nice to them. So thank you.
Yeah. And that's absolutely true, and you're very welcoming. And I've had people tell me just how nice it is to come in as a first time at a conference and be so warmly welcomed, embraced, and included. So thank you very much, and keep it up. Okay, what's next? Okay, it's good I've got a thing here. So this is the end of HTLV 2024. I know we waited 5 years for a face-to-face meeting. But you're not going to have to wait quite that long, hopefully, other biases permitting, to the next meeting. So I'm going to first of all ask Andrea to come and tell us what the next meeting will be chronologically, and it's in Europe.
Yeah. I would be very pleased if many of you come next year to the European meeting, the HERN meeting 2025, the HTLV European Research Network, and it will be in Erlangen in Germany at the beginning of July 2025. That's what we are thinking at the moment. So where is Erlangen? Erlangen is in the south of Bavaria, but not as south as Munich. It's close to Nuremberg. In Nuremberg, there's an airport, and you can be in Erlangen within 20 minutes by cab or 35 minutes by public transportation. And our institute is located in the middle of the botanical garden, and that's where we plan to organize the meeting. And since you're coming in summer, we cannot go to the Christkindl market, which takes place in Nuremberg. However, we have nice beer gardens and beer and pretzels, and I hope a very good program, and more information will come soon, and you will find it at HERN2025.net very soon. Thank you.
I always love the conferences where the selling point is the beer. Where's Leuven? Next up.
Okay. So next up is in Brazil. So I would like to invite you to come to Brazil, to the beautiful state of Maranhão, which is in the north of Brazil, northeast. Is a kind of colonial city, São Luís. The French will give the name to the city, São Luís, Saint Louis. And it's a beautiful place, colonial Portuguese. And we have this marvelous park of dunes and lagoons of not salted water.
So, fresh water.
Yeah, fresh water. It's very beautiful. So please come join us and share your knowledge with us. This is a very generous community. So we hope you came in October 25. You don't have the dates yet. Thank you for this opportunity.
More than beer, I like conferences that mention sand dunes and lagoons, so that's great. And a generous community, but finally, Amanda, are you going to say, would you like to invite people to the next conference and then we can all go home? One more, and then it's yours.
So I guess this is what happens when the person who's organizing the next meeting has to catch a flight. The second one comes up, so I'll do my best here. But so our next meeting is going to, for this one, is going to be in 2026, and we are going to be having it in the USA. Dr. Pooja Jain is going to be chairing this, and we're going to be doing it at the William F. Bolger Center in Maryland. I think Pooja described it yesterday at the general meeting as a resort type of experience. I think what she meant to say was there will be lodging and conferences in the same building. Maybe not the resort everyone's thinking, but we'll do our best. But these are just some pictures of what it is. And it's not here, but it's the Washington airport is very close, and then there's a train that you can take and transportation there. But yeah, we're looking forward to having everyone there, and we're excited. Thanks.
Thank you. Bye.